Hello everyone. Today we will look at structure of DNA, which is also known as deoxyribonucleic acid. So let us first revise a bit. So what is the function of DNA and where it is found? We know that every cell got its own nucleus. In every nucleus we will find something called DNA. Okay. So what is the function of DNA? Now coming to the function, if you have a basic idea of central dogma, you may know that when we have a DNA, okay, we get RNA from it by the process of transcription, then we get proteins by the process translation. Okay, so proteins have various functions in our body. Let us not go into the detail now. Now let me explain the nature of DNA. So you may all know that DNA are dual stranded, which means you can see in this diagram it is not one structure but it is a two thread like structure attached by one another. Okay, so there is hydrogen, these two thread like structure are attached with one another by hydrogen bonds. Okay, so if you have a little bit knowledge about music, you know that there are seven notes. There are seven notes. Okay, like sa, re, ga, ma, pa, dha, ni, sa. Okay, so by the combination of these seven notes, every music is created. Okay, in the same way, in DNA we have four types of nodes, or you can say building blocks. These are number one, adenine. Okay, then we have thymine, then we have guanine, then we have cytosine. Okay, so we got four building blocks. By the combination of these four blocks, we find various what to say various combinations of nucleotides in DNA. Okay, so let me bring one other image, okay, which will make you it little bit more clear. Okay, let me import one image. Okay, so in this image you can see, okay, there are two strands, strand number one and strand number two. Okay, so here are bases attached with one another. Okay, so here you find two types of bonds. Okay, for example, in this structure you can see this is cytosine and this is adenine. Okay, they are attached by bond which is called phosphodiester bond. Okay, now in this strand you can see adenine and thymine. This is thymine. Okay, so these are attached by hydrogen bond. These bonds are called as hydrogen bond, and this is called phosphodiester bond. Okay, now you can see 1 minute. Okay, let me clear up. Okay, so you can see, let me construct a DNA. Okay, guanine, adenine, thymine, cytosine, adenine. Okay, this is a construct. Okay, but it cannot randomly binds to any other simply any nucleotide. Okay, for example, guanine can't bind to thymine or adenine. Okay, so there is a universal rule. For example, guanine will always bind with cytosine and thymine will always bind with adenine. Okay, now you can see here when guanine, in the case of guanine and cytosine I have made three bonds, and in case of thymine and adenine I have made two bonds. Okay, this is also one of the other universal what to say law, or you can say whatever you can say. Okay, the guanine and cytosine will always form three bonds and thymine and adenine will always form two bonds. Okay, now let us construct the complementary strand. Okay, for example, this is guanine. Okay, guanine will always bind with what? Cytosine. Okay, so we will make 3 bonds here. Now thymine will always bind with adenine. Okay, we will make sorry, 1 minute. We will make 2 bonds. Adenine will always bind with thymine. So here we will have 2 bonds. Here thymine will always will bind with adenine. So we will have 2 bonds. Cytosine, guanine, 3 bonds. Cytosine, guanine, 3 bonds. Adenine, thymine, 2 bonds. Okay, and in between, let me change the color. Okay, in between we will have phosphodiester bonds. So this is the nature of DNA. Okay, we also got 5 prime 3 prime ends. Okay, it will be just opposite. Here it will be 3 prime and 5 prime ends. Okay, so I will explain it in some other video. Okay, so for more videos like this, please subscribe to our channel, and if you like the video, please hit the like button. Thank you for watching.